In today's episode, we'll be going through enzyme exam questions. The way I'm going to do this is that I'm going to show you from start to finish how I'm thinking, approaching, and answering the questions. Remember that the mark scheme is in the description box below. So let's begin. Describe how the rate of reaction was calculated. We look at where that's written in the table, and we can see that's s to the power minus 1. So you don't even need to do A-level maths, you just need to have done GCSE maths to know that if something is to the power minus 1, that is essentially the same as saying 1 over that thing, so 1 over seconds, right? So that's your answer. Next one. Explain what the standard deviation shows in table 26. What I'd like to do is straight away define what standard deviation is, as I know that always carries a mark. And I would be like, well, you know what? Standard deviation shows the spread of data around the mean. Now for the second mark, I would think back to the table and see what I can actually see from it. So looking at the third column, I can see that the standard deviations are very, very small. Remember, the difference between 0 and 0.1 is just 0.1, but the difference between 0 and 0.000 something something is very small. So just by stating the obvious, we can be like, all data have small standard deviations, right? Because they're not 0.1, they're much, much less than that. That's one thing I could say. Another thing I can be like, there's little variation actually in the repeats, because if the standard deviation is so small and so close together, then that actually shows that the experiment has high repeatability. Next one says, explain the pattern shown in the table using table 26 and figure 26. So we need to use both, and it's an explain, so you need to use your scientific knowledge of enzymes. Straight away, what I would always look at when standard deviation is mentioned in the question of the error bars, are they overlapping? In this case they are. Well, what does that actually show? It shows that if the error bars do overlap, it increases like the uncertainty of the data, right? And that's something you can say, because that shows that a higher concentration of bromelain, there's like more uncertainty. Another thing you could talk about is how the rate plateaus towards higher concentrations. And to back that up with scientific knowledge, you would be like, well, that's because there's like less concentration of substrate, or you could say that active sites are mostly preoccupied. Another thing you could actually talk about is the fact that as the enzyme concentration, being bromelain, increases, the concentration / like the availability of the substrate remains the same, because no matter how much you increase the concentration of bromelain, you can clearly see that the rate has plateaued. So those are two separate points, but basically under the same umbrella. Okay, and that's the end of the video. Remember, the two most important exam techniques are read the question first and state all the obvious points. Now over to you. Try some questions and let me know how you do.